Thanks everyone for coming to the Hungarian Innovators Club's event. We usually give our events once a month. Even under the pandemic, we did that over video, and nowadays we like to do it in a hybrid mode. Today, our presenter, Istvan Szanda, will be talking about nuclear medicine and nuclear imaging and things like that. Please help me welcome Istvan Szanda.
Thank you very much. Thank you so much for the opportunity to meet you and share a few things. I've just moved to the Bay Area recently from Washington, D.C. I used to live in London, UK. I certainly like the sunshine here and also the people. So I have prepared a slide, but I wasn't on my own. I would like to mention Peter Kontz, my colleague, who is an application specialist for Medizo, who did a great job helping me with the slides. So, nuclear medicine, uniquely Hungarian science. I'm not a medical doctor, and I don't particularly like blood either, so I just warn you there's only one slide that can be a bit disturbing, and this is the next one. Because we want to talk about the why of, it's not the goulash yet. We want to talk about the why. So why medical imaging? Why nuclear medicine? Why we bother? So if you put on YouTube brain tumor detection, it's a snapshot of a 1933 video. So that's obviously somebody's brain. The scar was removed. Everything else was removed. And, you know, a couple of decades later, if you, again, choose brain tumor detection, that's what is happening in a hospital. Instead of a complicated operation that is already risky, like opening somebody's skull, it's worth, if you once cannot sleep well, it's worth watching this video, how gruesome and how terrible diagnosis was 100 years ago. And now it is a matter of minutes, no pain, and you get an image of the whole brain with much greater accuracy. So when we talk about medical imaging, we don't just talk about one type of medical imaging, we talk about a few, and we will talk about them today. So one of them is FDG. You can see a patient here, you can see parts of the gut and the brain and the bladder. We can also think of CTs or CAT scans. I'm sure some of you have already had maybe bones fractured, broken, and you had to go to a CT scan. We also have MRI, typically for joints, brain, soft tissue, and also have SPECT. SPECT is similar to PET, and I will discuss what sort of imaging modalities these are, because the PET and the SPECT are the primary nuclear modalities. The other two are also helping us to do great nuclear medicine. So why molecular imaging? Molecular imaging is non-invasive. Just think of the first slide. It also works on living subjects and intact subjects. It's greatly appreciated if you do, for instance, drug research. You don't need to sacrifice the lab animal, take the organs out. You have an intact subject. You can also image real time and over a longer period of time. You can use the same subject and follow it up as you do with humans, especially after cancer treatment, disease progression, Alzheimer's, and so on. We are working on the molecular level. We target certain receptors or certain parts of the body by a molecular target. So we really track molecules in the body to that level. And we can see the big picture. You know, that's one thing. If you have a sponge cake and you cut it into half, you want to pick either the cake itself or the breadcrumbs or the chocolate or the raisins. You don't know, if you can only pick one, whether it would be a chocolate, a raisin, or the cake itself. It's quite important, especially for brain imaging. I will tell you an example later on. In order to see each tissues, we want to see them in situ, similar to a goldfish. If you investigate a goldfish by taking it out of the aquarium, it might not have the same result if they are in their natural environment. We can do all of this with nuclear medicine. And we can also follow not just a snapshot of what is happening, but follow everything up as a movie. So 2 breakthroughs in medical imaging. First was the first X-ray. It's not quite a CT yet. It's an X-ray. It's 1895. And 100 years later, the first FDG PET. So X-ray was certainly the first modality in medical imaging. And 100 years, all the molecular imaging techniques came to fruition by inventing FDG. I will talk about what FDG is. You see the heart, you see the bladder, you see the brain, some of the background, some of the tissues around, an anatomical modality, and the functional or molecular modality. So if you talk about PET, positron emission tomography, we have PET because we have FDG. FDG is a type of a glucose that goes into the cell because the cell sees it as food, but cannot digest it. So it gets trapped in a cell. It's a glucose, but not quite a glucose. In 1968, that was in Czechoslovakia, a couple of scientists synthesized this. And in the US in the 1970s, they started using it for clinical imaging, for human imaging. So what you can see here, the brain on top, you can see the tissues, you can see the bladder, you can see the kidneys, parts of the body in image A, and you can see a lot less in image B. That's the power of disease progression and follow-up. It's the same patient, but probably after a chemotherapy. What you see here on A and you cannot see on B, those are the cancer cells. And we can do this imaging in a matter of maybe half an hour with reconstruction, covering the whole body, having the subject as a whole body, intact, no pain, minimal risks. And this patient is very lucky because you can see parts of the kidney, you can see the bladder, you can see the brain, but pretty much all the other cancer cells are gone. So this is a very positive story for the patient. Again, if you think about the diagnosis without imaging, that would be a lot more difficult. You wouldn't be able to count everywhere where the cancer is. You can only rely on the symptoms or just pushing down and feeling the anatomic parts. Here you can go down to the molecular level. You can see molecules here, not just cells, molecules. There's another application of molecular imaging, and that's drug development. So this is a fantastic slide from 2022. You can always see the references here of the drug discovery process. You have 200 candidates, and in order to get one approved, average takes 15 to 25 years. And you can see a significant cost of the approval process. This is why pharma is a risky business, because you have to have a lot of molecules and spend a lot of money to have one or two in the pharmacy later on. In this part, after all the chemistry is done, we speed up, and that's one of the side of our business, speed up preclinical testing. Preclinical meaning not yet human subjects. In this case, lab animals. We can speed up the discovery here to be translatable to humans. So the idea is to stick the same syringe in. Whatever we do with animals, we can also do with humans, because we have CT on both sides, PET, SPECT, MRI on both sides. It's about the drug discovery. And drug discovery is an extensive, expensive process involving various fields from molecular biology, chemistry, physiology, doctors, physicists, engineers, chemists, genetics, immunology. It's a complex topic. It starts with a decision, what sort of pathology you want to address, then finding the molecular targets, and step 3 is finding the modality that can enable you to see those molecular targets. Then chemistry, to make sure you have the molecules ready. Labeling, meaning labeling, adding radioactivity that we can track. I will talk about it later. And then molecular biology, small animal, large animal, and imaging, analyzing these images, and then we can translate it to clinical imaging. So how can we choose all these modalities? These modalities are like senses. Some of the modalities are good for processing of very finite, very small information. Some are very sensitive. Some provide detailed information of the body. Some are more molecular. So spatial resolution is one consideration. Spatial resolution is the resolution of how good the eyes is, how small can you see a certain target. Sometimes it's important, sometimes it's much less important. There are other considerations. Sensitivity. How many moles, so what is the number of molecules we can see? And you can see in terms of MRI and PET and SPECT on a logarithmic scale how different the sensitivity can be. And certainly the champions of sensitivity are the nuclear modalities, so PET and SPECT, because you can go down to even 10 to minus 15 moles, that sort of a sensitivity. So if there's a molecule somewhere, we can find it with that level of accuracy. We also have sometimes a need to have a dynamic information. For instance, we want to do drug research, and we want to see where the drug goes from moment 1 to maybe in 1 hour, 2 hours, 3 hours, to target these pathways and to follow them up. So sometimes it's great to have dynamic imaging. Again, if you think of CT, some of you, I'm sure, have gone to a CT exam and they gave you contrast agents. So that's a way to aid this kind of a dynamic or temporal resolution to see where it goes. You see the blood flow, typically with a CT agent. With molecular techniques like PET and SPECT, it's not just the blood flow we can image. We can image to the receptor level or a much more like other pathways, not just the blood. In terms of quantification, that is a great advantage of the molecular modalities because we know exactly the number of molecules we injected, number of radioactive targets we have, radioactive tracers we have. We can quantify the amount of uptake in a certain region, whether it's the heart, whether it's the brain, whether it's tissue, anywhere else in the body, or cancer cells. So you can, if you think of the follow-ups prior 3 or 4 slides prior to this one, it is about the quantification of the disease progression. Also, with advanced technique, we can have mathematical models of a drug, pharmacokinetic modeling in a mathematical way, in a quantitative way. So that's how you can do it. This is a liver. If you just look at the images, it's very difficult to see the difference because it's all about the grayscale. If I move the grayscale here, you can create an image for whatever you want. It's like Photoshop. But if you are into quantification, you can see the maximum uptake in the organ has increased or changed. And we can compare different patients. If you do a drug research, you have a cohort of patients, you can quantify the impact of a drug, the effect of the drug as well. I want to talk about tomography for a second. So tomography is like slicing a salami. You have the slices, but it's in the inverse way. You have the slices, 2D, and you want to put the salami back together. So that's tomography. We have projections. The slices are called projections, or sometimes called slices, depending on the modality. So we have the slices. These are usually cross-sectional. So slicing this way, like how you slice a salami along the axis. And we can create the volume back, typically these days via computers, to have a whole body 3D map to find whatever we want to find. So tomography is the 3D way of doing imaging. And PET is a tomography. SPECT also is a tomography, computed tomography. MRI, they don't say tomography, but it also creates 3D volumes. However, we don't have slices the same way as with the other modalities. So now we understand why. Now we understand the use. We understand tomography. Let's see about what the nuclear part comes in. So just if you have had your physics classes maybe some years ago, there are different forms of radioactive emission. It's similar to firearms. There are different calibers of firearms. Some are small and does not penetrate very much. You know, if you think about a tank, it has a lot higher caliber that penetrates a lot. So there are different forms of radioactive waste. Alpha decay has large particles that they do not penetrate a lot. Even a piece of paper can stop them, but it's quite destructive around. It's important to know, because I will talk about theranostics a little bit later, about how we can use alpha particles to cure disease. So we have something that is just in the vicinity, but is lethal. If you think of that Russian spy who was killed by polonium, you know, the coffee a couple of years ago, he was in the UK. It was an alpha emitter. So you could not detect, even with a Geiger-Müller counter, the radioactivity. But when you drink it or it comes to your body, it does a lot of damage. So that's alpha. Beta, we don't talk about too much about beta today, but there's a special beta emission as this positron. Positron is like an electron, but an alternative universe. So it's antimatter. I know it sounds it's science fiction-y, but you know, let's assume there's an alternative universe somewhere, and where every electron is a positron, so they are charged differently, and every proton is an electron, so charged differently. So we have the beta particles, and gamma penetrates through a lot. It is not as sort of harmful as alpha. It has much less kinetic energy, but can penetrate a lot of tissue. We use this in imaging. They are all in the electromagnetic spectrum. The gamma rays are here, X-rays are here. Then we have a little bit of the visible light, you know, heat, microwave, radio frequencies. We use quite a bit of the electromagnetic spectrum, radio waves in MRI, X-rays for CT and X-ray, and gamma rays for tomography of SPECT and PET. These are the nuclear techniques, so they are operated either in the X-ray and the gamma ray level. So just a few words about computer tomography. You have the object here, you have the X-ray source. The X-ray source is moving around the object, creating these slices, so these 2D projections. And the more projections we have, the closer we are to the real image. So you can see a reconstruction, meaning we have these projections together, we spin them around, and you get, for a few projections, not much, but if you have enough projections, you can get quite a good image. So that is called reconstruction. So we have 2D images, we reconstruct them into a 3D volume in tomography. So that's how it works with a CT, a humanist or an animal. is lying there. We rotate the detector around with the source and collect these 2D images, then reconstruct them to have a nice human CT. So it was the first CT from Hounsfield in 1969. It was a brain and a little bit of a rotation, but not too much. You could see that it was able to rotate to collect images. So from this image, within a couple of decades, we went to very, very nice CT. I promise you to talk about positrons a little bit more. Positrons are like electrons, but opposite charge, and it is a form of an antimatter. So what we're essentially doing with positron emission tomography is we chase antimatter in the human body. This antimatter does not live very long in the hostile environment, so the matter environment. It annihilates, that's the jargon for it, and creates two of these gamma rays. If you think of gamma rays, those are the ones that can penetrate. And this is what we want. We can detect these gamma rays around the subject or an object by collecting them. How do we collect them? We have the gamma ray, a scintillator crystal. It looks like, I will show in the next slide, it looks like glass. It absorbs the energy of the X-ray and creates a light flash. With light, we can either, using a photomultiplier or a silicon photomultiplier, we just detect the light and create electronic signal from it. With that electronic signal, we can use the position to create those slices, and from the slices, we create the whole volume. So we have an absorber of the gamma ray that creates visible light, and then we work with light. It's very easy to work with light. You know, we have the old televisions. If you remember, some of you remember of the old TVs, they were vacuum tubes. We use the same technology as the other way around. Instead of having an electronic signal and create light as an output, we take light as an input and create electronic signal as the output. So that's the photomultiplier tubes. So this is how the crystal looks like. It's not just pretty, it's expensive too. So this bit is about $35,000. Fits my palm. We have the photomultiplier tube. It's similar to the old TV I mentioned. And we, you know, with the electronics, we create a module using the crystal and the PMT, and we put the PET ring together to be able to see the object from all sorts of angles. We want a very good angular sampling, meaning you want to have a lot of slices around to create a good image. So that's how it works. We have the human here in this case, and all these antimatter containing molecules, tracers, they don't contain when we inject, but soon after they decay. And then we detect the possibility of a certain decay happening in a certain position. So with the human, the more decay is detected from a certain region, we can safely assume, because we gave the patient the radioactivity, the special radioactivity, that where we detect most of these, that was the place for the uptake. And where the uptake was, the uptake correlates to the function. Let's think of FDG for a second, because then I promise from now onwards it would be much easier. I know there's a lot of information. So let's think of FDG. FDG is a type of glucose. We labeled it with radioactivity. We inject the patient with glucose. Every cell likes to eat. Every cell can take up glucose. But certain cells have a lot of appetite. Typically our brain, and typically cells that use a lot of energy. And reproduction takes up a lot of energy. Cancer cells reproduce much faster than any other cell. I'm sure there are biologists here who will correct me that it's not true, but to make it simple. The more appetite a certain cell has, the more uptake it is, meaning they want to bring the food in more and more of these almost glucose molecules, but they can digest them, so it gets trapped in the cell. It will be excreted later, but we want to image the patient where there is an accumulation, an uptake of this molecule, and have that snapshot. We can also do the movie, but for FDG it's typically a snapshot. And the way we do it is, statistically speaking, we collect where these nuclear or radioactive decay events happen, and then reconstruct the volume, reconstruct the human body. SPECT is very similar to PAT. The only difference is here we don't use any sort of antimatter. We use just gamma rays and collimators. These are pieces of lead or tungsten, work similar to the camera obscura. So you have an object and it has via some optics that is called a collimator, it has a projection somewhere, and we use these projections again to reconstruct the 3D volume. So PET and SPECT, they are very similar in terms of mechanics. PET uses antimatter, so we don't need collimators. We have more information from the time. With SPECT, we use collimators. And similarly to PET or CT, we collect information from as many angles as we can. Usually in case of SPECT, some rotation is involved. So we rotate around the patient. In this case, it's a mouse. We have the bladder here. We have the gut, a bit of the brown fat tissue, the salivary glands, and the thyroid. This research was based on thyroid.
Which one of these had that sensitivity of 10 to the minus 15?
PET has approximately 100 to 1,000 times more sensitivity, but SPECT is not far. It's a great question, not far below. So we are still on the molecular level.
So with 10 to the minus 15, that would be about 600 million molecules.
MRI is slightly different. I don't want to spend too much time on MRI because it's not the main focus of today. But there's MRI. It works by having the water molecules or protons in our body aligned with high magnetic field. And then we create resonance with these protons. And then from that resonance, we give an electromagnetic pulse, and then detect one. So it is a radio, essentially. We are radioing to our protons. Probably the quickest explanation I can give. So it does not work the same way. We don't need to inject anything to the patient because the water is there. There are some MRI contrast agents, typically gadolinium or iron oxide, whatnot, but it works a different way. And why do we have MRI? Why do we have CT? Because they are great for anatomy. You see where the kidneys are, you see where the heart is. With CT, you can see a lot. It looks a lot better on my screen. With a CT, you can see the bones very well. CT is an absorption. It's actually detecting the shadow. So you have the X-rays, they get absorbed in the body, and the leftover is the shadow. You reconstruct them. And if you combine anatomical modalities like MRI and CT with PET, you have a very powerful tool. So how does Hungary and goulash and everything come together? So I want to focus now on the business aspect of the story. So there's a company called Medizo. It's a group of companies right now, 8 companies together. It was founded in 1990. And we have installations of clinical and preclinical scanners or cameras in 100+ installations. So we have a lot in North America, Europe, Africa, Asia, as well as for the animal cameras all around the world. We are very proud of Medizo because it is a Hungarian company. We do research and development in Budapest, as well as manufacturing and all the rest as well. So we are not part of, it's a privately owned company, and we work with actually probably all of our colleagues, at least in the headquarters in Budapest, Hungarian. So we had Gamma to build upon. Gamma was founded in 1920, and I'm sure you have seen one of these old Gamma type photographs, cameras. Pajtás, that was the name. So Pajtás was a Gamma product. And that was nothing to do with nuclear medicine at all. In the meantime, George Hevesy in 1943 got the Nobel Prize for the tracer principle. I will explain it very soon. And a little bit later, because the initial SPECT camera was a gamma camera to detect the gamma rays, was invented by a Turkish-American electrical engineer, Chicago, called Hal Enger. Soon later, all around the world, people started producing this device. So the idea was coming from George Hevesy, and then Gamma started focusing on these devices, these radiation detecting device for use. A lot of years later, Medizo was founded. Actually, Medizo was founded by one of the Gamma engineers, Istvan. And since then, Medizo has been on a roll with several successful campaigns introducing different products, not just clinical, not just human use, but also preclinical use, and also offering clinical services. So who was George Hevesy? He got the Nobel Prize in 1943 or I think it's chemistry, right? Chemistry, because of the tracer principle. So if you think of the title, that was slightly provocative, I must admit, about the goulash. So how come this gentleman, George Hevesy, discovered the tracer principle? It's quite a remarkable story. So he was working in the UK and was not very happy about the food. I don't know whether you live the UK or not. I did for 10 years. So he was not very happy with the food. And he accused the landlady of sort of, recycling the meat from the previous year. So last night's hamburger can be minced by the following day. Obviously, the landlady vehemently did disagree with all allegations and denied all allegations. Already, George Hevesy was given a task a couple of years earlier in the UK by his supervisor to try to have lead isotopes that are radioactive and lead isotopes that are not radioactive separated from each other chemically. This poor guy eating, bad food and not liking the UK very much was given an impossible task because he worked for 2 or 3 years trying to separate radioactive lead from non-radioactive lead. But it is impossible, completely impossible. So he was very upset with an upset stomach and had the idea of using radioactive lead in the burger and have a radiation detector the following day. If the mince pie the following day was radioactive, obviously. It was his lead from the day before. So he got the Nobel Prize because of his discovery of the tracer principle. And that's the key. You cannot separate chemically the radioactive and the non-radioactive isotopes. This is why we are here. This is why we can do nuclear medicine. This is why we have such a strong diagnostic tool or department instead of just cutting everybody open to diagnose in terms of the molecular level, because of this, appetite and being homesick and craving for a good food. So he started working on that, and that was a lead 210, so the isotope is 210, in 1920. So he worked on this tracer principle with plant first. So water the plant with lead and could take images of where this lead went. And that was the invention that led up to PET, SPECT, tomography, and molecular imaging. He also had another interesting story. He was working in Denmark in 1943, and Denmark was occupied by the Nazi Germany in 1943. 2 years prior to that, German scientists sent him and his lab the Nobel Prize. That was gold, but it was a crime against the state to export gold from Nazi Germany, so led to an execution. So what he did when Germany occupied Copenhagen is he had some aqua regia and dissolved the Nobel Prize of the German scientist who had sent him the prize itself. A couple of years later, although the lab was not intact because people were searching the lab for precious metals, nobody bothered to take the aqua regia off the shelf. It was intact. So he could get the gold back, sent it to Stockholm after the war, and the Nobel Committee agreed to recreate the Nobel Prize for those German scientists. So he was a really funny man in that sense, and a very ingenious man. So just to go back to gamma művek a little bit, that's the other aspect where we are here, is the Hungarian connection. It's the camera between 1955 and 1962, the Pajtás. And the profile changed in the 60s. In 72, this was the first gamma camera. So it used SPECT style. It was not tomographic yet. You see, there was no rotation around, just the patient lying there and see what is going on in a molecular manner. So gamma művek existed till about 1998. 1998, I think 7 or 8 Medizo bought gamma művek. And this was not the first Medizo factory. This was the first Medizo factory I started. So it was in Újpest, quite a rundown building. The managing director had this fantastic yellow Suzuki, I believe three cylinders, maybe 4. We had all the equipment lying in a line. It was very difficult to navigate in between the two. So I was till about 2012 or 2013, and this is the Medizo headquarters now, 10,000 square meter in Óbuda. We have a huge facility, so the production is here, R&D is there, and we have a large warehouse, and we have been, since really the foundation, quite successful. This is where we are. This is the Hármas Határhegy, so one of the mountains in Óbuda, and the factory building. A small booth in 1993. So the gentleman with the mustache, so gentleman mustache 1, is the founder, Istvan Bagaméri, and the mustache 2 is founder 2, László Nagy. Actually, Istvan Bagaméri and his wife established the company because he was working for gamma művek. and saw the opportunity to bring the technology, bring the know-how, and establish his own company. This is a couple of years later, a lot larger booth. To date, we have the largest booth during the ENM, this is the European Nuclear Medicine Association. A nice story. A couple of years ago, probably about 18 years ago, we had this brain camera to detect disease, again, molecular, in a molecular way of the brain. And a German/American company saw the opportunity to use the same exact machine and create the small animal scanner. Instead of humans, we could now image mice and rats for the translational research I was talking about, for the drug research. Istvan Szanda got the prize for this innovation in 2006. A couple of weeks ago, although there's no mustache, but the English, Istvan Szanda's son, Gergő, also got the same award for the MRI. So now Medizo, although the background was in SPECT, now have CT and SPECT and MRI. So it has been a story that is a story of success, story of Hungarian innovation. From the concept till to date, we keep innovating, bringing new products, couple of awards. Obviously, the list is rather short. On the slide, we have the clinical line, we have the preclinical line. Obviously, we are not on our own. We compete daily with GE, Siemens. Philips used to compete with some of the smaller companies in the preclinical field. It is a tough business, I have to say. I'm the managing director or CEO in the US and Canada. So it is a tough business, but we can make it. And I want to serve as an inspiration on behalf of the company for a small Hungarian family-owned business, quote unquote, conquering the world and changing the field of a very special Hungarian field that is molecular imaging. That was still based on George Heves's work. So for the leftover about 15 minutes of my talk, I would like to show you a few applications. How this nuclear imaging, we have discussed the science field, we have discussed a little bit of the anecdotes and the historical aspect and also how we can help people, how we can help industries with this technology. So this is cardiac imaging with SPECT. You understand SPECT very well. We inject radioactivity and we take images of the heart. So you see the slices here. Obviously, I don't have a 4D monitor to show you a 3D. So these are slices of the brain, slices of the heart. And between stress and rest, stress is when somebody is doing some exercise. So there is an increased need to get blood flow in the heart. You can see the defect. This is a great way to diagnose an ischemia or after a myocardial infarction, the dead tissue. Because you see clearly for a healthy heart or not without the stress, so it's a resting state, that the coronary arteries can supply the heart with enough blood, but in case of stress, it's typically like a treadmill or walk, depending on the patient, there's already a defect, so there's not enough blood flow. So we can image the blood flow with SPECT. We can also image the bone, especially the bone repair. So we have this molecule here, we inject to the patient, and you can see, unfortunately, some people have bone mets, so metastasis. When a bone is trying to repair itself, you can image it, also infection, healing, or after a fracture. So not just the heart, we have a specific tracer for the bone. Again, this image takes maybe like 20 or 25 minutes with no pain. Somebody is lying there. We scan get the whole body, and to a molecular level can image the bone. This is renography, so kidney images. After a kidney failure, some kidney disease, you can compare the uptake in one kidney or another kidney. And I wanted to put it here because you can see the uptake we were discussing, so the nuclear radioactivity concentration in the organ over time as well. So you don't just compare the two kidneys as a snapshot, you can see them as a movie over time. I've mentioned quite a lot about the PET tracer, so FDG, the glucose analog. This is how it looks like. It goes into the cell, but unlike normal glucose, cannot be eaten, cannot be metabolized. And you see a treated or an untreated tumor in case of this, probably a rat or a mouse. You can compare, even within the same animal, you treat one half with the drug, another half, just leave it as a tumor, and you see the disease progression. Unlike it's an MRI, it's very difficult to see because you see the anatomy. I said a little bit more water there or whatever, but with molecular techniques, you can compare in a quantitative manner. So you can count the radioactive molecules in this. So this is cancer neurology. I will show you a few neurological examples. Anything, brain eats a lot of sugar, or our brain. So wherever certain parts of the brain are active, they consume more sugar. So you can image the appetite of the brain. Cardiology, one of those, the heart muscle also takes up a lot of energy. So we can image that. Infection is also when there's an infection, the body is active around. So we can image that. And one of these typical examples is inflammation in a joint. Otherwise, quite difficult to image. We can do it. So it is from UC Davis. And I will play this video probably a couple of times. A couple of years ago, they built a whole body PET scanner. So you can get within a minute, and that's probably where the industry is heading at the moment. You can have in one seating a whole body imaged with PET. So you can see the injection right there. Up. You can pop up. So you can see, I need to be more precise. So you can see the injection, the line there. It reaches heart and lungs. It's quite a challenge to do it, but I don't give it up. I just do it here probably. Okay. So you see the line. It reaches the heart. You see the vasculature. Mm-hmm. So now the sugar is everywhere, and the uptake is happening after a certain period of time, because it started the detoxification by the kidney, and you see the cells with a lot of appetite right there. I think it's a pretty brilliant way to explain what is happening. Thanks to Simon Cherry. Simon Cherry, the supervisor, was my supervisor during my PhD too. So that's common with Simon Cherry and I, although I left science quite a bit these days. So Alzheimer's disease, it's another interesting, well, it's a very sad disease, but it's interesting to image Alzheimer's disease with PET. So these amyloid plaques, so misfolded proteins between the neurons, and the neuron communication breaks down. This is why there's a cognitive decline with Alzheimer's. You see a healthy brain, and you see a positive scan, meaning a negative scan, meaning a disease-affected brain. Small amount of amyloid plaques, a lot of amyloid plaques. How can we use it? So you can diagnose Alzheimer's before you have cognitive decline, because you do the imaging of these plaques, and that's one neuron. That's the size of what you can do. So you can prepare a lot more in advance, prepare the family, do some treatment, not yet a cure, but treatment of Alzheimer's. This is Parkinson as well. So in case of it's a dopamine receptor, so you can image the brain, a normal brain, but the Parkinson's brain, you see this region, how much less of an activity you can detect. So you can quantify, see the disease progression, and helps to find a cure as well. The next slide is an interesting application. We deal with that too. And there's one of my colleagues went to, it's in the UK, it's a Medizo camera, went to install one of these equipment in a veterinary clinic. And the horse was not too happy about the examination, and horses tend to kick around. So they kicked the $150,000 equipment. The service engineer who was doing the installation was quite unhappy about it and mentioned that, how come that a horse can kick that equipment? But the vet told the service engineer that the horse cost 6 times more than the equipment. So instead of worrying about the equipment, maybe we should have it softer so the horse does not have any injuries from the kick. Race horsing is still a big business. And one of these companies, within the group of companies, Bartek Technologies in the UK, is heavily involved in the Cambridge area of racehorse insurance work. So when you buy, an expensive car, you want to make sure that the paint is the same width. The paint has the same width all over, because there was no insurance, there was no accidents or collisions. People do it with race horses too. And nuclear medicine, molecular imaging is one of the ways you can make sure that the joints of the horse are healthy. For instance, the race horse is very important. That's another study. It's a little bit of the MRI. In this case, we can detect the brain regions that are active because of the blood flow. So you can highlight with fMRI, so functional and functional MRI, the active brain regions. So one study we have used is was with burgers I used to work for King's College London. It was the Institute of Psychiatry. We had 2 groups of patients. One group was obese, and the other had a healthy BMI. And their brain responded in a very different way to seeing a juicy burger. Now I'm hungry, I'm salivating. So I like this burger. My brain is all over the place trying to get active. So I would take a bite, but it's too deep. So their brains responded very differently to the visual stimuli in form of an image. And their brains, so people, statistically speaking, people who were obese responded similar to alcoholic people respond to the smell of a good whiskey or a bad whiskey, depending on the stage. So they were more addicted to it than people with a normal BMI. Also, that's a very important study about mirror neurons. They, in 1990, discovered mirror neurons in the lab between monkeys. So there were 2 monkeys, and they had their brains wired with electrodes. One of the monkeys was eating a banana, but they realized that the other monkey started salivating too, without eating the banana. Started to have the same regions active as off with the monkey who was eating the banana. I'm sure we have the saying of monkey sees, monkey does in the human scale as well. So these kind of mirror neurons are really important for imitation. And there is one particular, that's the politically correct way to say, people who are on the spectrum, so people who are affected with autism, they have a broken mirror neuron system. So one of the reasons who they don't perceive other people's emotions, they have that sort of a lack of empathy, because empathy is linked to mirror neurons as well, and social skills and so on. So we could, with fMRI, be precise to detect autism and to visualize it on this level. It's also an interesting study with a Buddhist monk and mindfulness, whether mindfulness is just pseudoscience or really work. They have done this study in University of Wisconsin Madison. He's probably the happiest person on the planet, or one of the happiest person on the planet. And also, meditation, mindfulness or meditation. They were following the brain of this monk and a group of people who were taught how to meditate over time, and visualize, again, on the brain part level, in the amygdala level, whether it had any positive or negative impact, and what specifically, meditation or mindfulness. And they found, yes, it did, and also improved between the amygdala and the prefrontal cortex, the communication. Language studies, we all speak probably more than one language. I only speak one, it's Hungarian. Very interesting to see how the brain is affected and how different languages, if you speak more than one, are stored in the brain. And bilingual people's brain work very different to people who only speak one language. So this Broca area is in charge of the speech function, and when it's damaged, it causes a speech impairment as well. So they have done this study with fMRI, and they found that bilingual individuals exhibit greater activation in this Broca area, and when you switch back and forth between languages, you can have, statistically speaking, less of a cognitive decline later stages of life. So it's very good to speak more than one language. I think it's one of the advantages of being an expert. So translation imaging, how can we get some of these wonderful tracers I discussed about? We need usually lab animals to do that. So we have the animal models in R&D, drug testing, and so on, and to translate it to clinical practice. Obviously, it comes at different challenges, because a mouse, in terms of volume, is about 2,000 times less than a human. And we still need to build the same equipment we build for humans. For something that is, a mouse is about 20 or 30 grams. A human is, 70 kg, or probably the average is a lot more. Maybe in Indonesia it's 70 kg, but certainly not in the Western world these ways. So we have to be able to have both machines, and we actually have a different resolution. It's a resolution question of how small can you visualize, and we do it very well. So it is zirconium, it's one of those tracers for cell tracking. So you see with great level of accuracy even the joints of the shoulder joints of a mouse, and you can even compare one to another. So you see how after 30 minutes, 2 hours, and 7 days, all the zirconium pretty much went into the joints and the bone. So that's the power. It's the same subject. You don't need to do any gruesome procedure, sacrifice any animal. You have the same subject as they were human, imaged over a long period of time. We can also do the same with heart. But a mouse heart is tiny, like less the size of my pinky finger's nail. And you see not just the left ventricle, but also you see the right ventricle here. That's even smaller. And we can do the same as we do with humans, analyze the function. This is one of the studies. So the last few slides are studies from the US. So these are our users, our customers, use Medizo machines. This is, again, zirconium with the GlaxoSmithKline for COVID research. So I can't tell you which vaccine, but they were tested in the preclinical stage and using the Medizo machines as well. So we are also DoD contractors, Department of Defense contractors as well. They do a lot of vaccine and infectious disease imaging. So Medizo machines. This is also a Medizo machine, SARS-CoV, but from Texas Biomed. It's again a military site in San Antonio. We work with TB research. We work with Bill and Melinda Gates Foundation. I don't know whether they separated the names or not. It's still Gates Foundation. So back then it was Gates Foundation, and they were happily married, assuming. So they bought 3 of these equipment from Medizo to work with TB and eliminate TB. It's not an issue in North America, but it's a big issue still in Africa, and people die because of TB. So we have these machines in laboratories, biosafety level 3 and biosafety level 4. Biosafety level 4 is the really nasty stuff, like Ebola and uncurable disease. So we have these machines in those environments, looking for cures for infectious disease imaging. Again, monkeys have very similar brains to ours, so we can have the Alzheimer's studies of different regions with and without diseases. It's a dopamine transporter. I think it's Parkinson's, not Alzheimer's, because that should be a dopamine transporter. However, you see the level of accuracy with anatomical modalities, as we discussed, that's an MRI, and functional modalities, and then combined together with great precision. A mouse vertebra with this level of accuracy, and the mouse liver, okay, using some contrast agents, you can get that resolution as well. And the rat angio, you see even a rat, how many vessels they have. And you can see even the smallest ones. So an unusual application, moving on, the last few slides, is carried out by Virginia Commonwealth University using Medizo equipment for nuclear reactors, nuclear plants. So it's nuclear, again, nothing to do with nuclear medicine, but we use nuclear medicine or nuclear medical tracer principles to have engineers information about how to design the cooling system of these nuclear plants. Why? Because you have to deal with a lot of heat. So the cooling should be continuous and the flow should be laminar. Because if there's a little bit of a non-linearity in the flow, it can overheat the tubes. So they use this equipment. You can't really see because it's under this cover. This is the equipment from Medizo. And they build up a tubing system, and using the same tracer principle George Habashi used with hamburger meat or reused meat and plants, and we use for Alzheimer's diagnosis, they used to detect the particles while being in a flow to make sure the flow is nice and laminar. places or wherever they are. They know more about this than I do. Maybe laminar is not the right word. It was a long time ago when I did fluid dynamics at university. I'm a physicist by training. So you can visualize using the same tracer principle the flow. And we are looking into other applications where flow is quite important. One of them is chocolate or ketchup or anything where you have a fluid going through a tube. You can visualize using the same tracer principle and visualize the flow. And so what we do as image analysis, so you have these pretty images, we can draw regions of interest and see the uptake, see the radioactivity in these regions of interest over time. So if you are planning to change careers, be an imaging scientist, most of the times you deal with regions of interest and time activity curves. You see the activity over time. We can move these images if you have more than one modality like CT and MRI, PET and SPECT. You can fuse them together, visualize them in different ways. This is how a program looks like. It is used clinically and preclinically as well. In this case, it's a preclinical equipment. So you see a large portion, small portion, other portion. You can compare more than one. There's a lot of image analysis, creating the radio kinetic models for a tracer. So this is raclopride. It's a brain tracer as well. Cell tracking, you can get down to that what you asked, very, very small amount of molecules. Even 17 days later, you see the cells over time. This is just a rabbit head. Some rabbits are used also in research, so not just mice, rats. This is the marmoset lung. So NIAID is also using Medizo equipment for tuberculosis, COVID research, and NIH, including AIDS imaging research. We have quite a large install base. So animals lie in these beds. They are all heated. They are very well cared for. We want to eliminate any potential stress because stressed animals' physiology change. So they are all heated, luxurious, more than one top of each other, top floor, ground floor. One of the challenges is animal welfare these days. A lot of publications were published and a lot of sites. So I'm just a few minutes after the hour, but I thank you very much for the attention.
We had a couple of questions already from the remote audience.
Sure.
One of them was, what size of hospital does your company target? You have a healthcare network, smaller labs or smaller clinics, or what do you have a target size?
So in the US and Canada, we only deal with preclinical, so lab animal equipment, all over the world, from doctors' offices to health networks. So Medizo's revenue is half clinical and half preclinical, half animal research, and typically Western countries buy from us because animal research and drug research generally, I mentioned how many billions of dollars it costs. So it's typically, US, Canada, Germany, UK, but we have clinical cameras in the UK, in Germany, a lot in Turkey, and the ex-gamma MUVEC, so prior to 1990, that region as well, from Ethiopia to, we have over 100 countries.
The next question was, are you able to show the absorption process of medication in the respiratory system as well?
Great question. There are different ways. One of them is technigas. So a patient inhales this technetium gas and then see which part of the lung it absorbs and which part does not. Some of these, so it depends on what you want to see. In this case, it was more about the scars, I believe, after an infection. So every infection leaves scars, and the lungs are no different too. So this is about lung scar imaging. There are different ways. So everything, the scanner is the same. The question is whether you have the molecule for it or not.
Another question was, where in the San Francisco region is your office, R&D, manufacturing, or sales in the US?
So the R&D and manufacturing is in Budapest. Our office in the US is in Arlington, Virginia, because of the military and the government, NIH. And the San Francisco office is right now where I am. So right now, right here.
And one more question I see. Does patient motion during the scan pose a significant problem?
Great question, and it depends on the length of the acquisition. So it depends on the length of the study. Obviously, if it's heart motion, we don't really want to stop the heart, but there's gating involved, so you can detect and record the signal, the ECG, and use it for imaging, similar to respiratory motion. Animals breathe differently than humans, especially under anesthesia, because while you can tell a human to stay still for a little while, you cannot really tell a mouse, so they are usually under anesthesia. And we do gating too, so only if it's critical, if it's like an orthotropic tumor somewhere, we can virtually stop the breathing, so we can only use that part of the acquisition where there's no motion. And patient motion, yeah, sometimes they feel uncomfortable, especially the MRI. It's really loud. It's claustrophobic. So you have to correct for it one way or another. It's another field. I was trying to. It was quite a challenge to put this presentation together. There was quite a lot of physics, biology, chemistry, medicine involved, and that's a whole field of patient motion correction. Best is if they don't move.
One question here. So how do you compete with the competitors? Your technology is not unique in a way. So how do you position yourself competing against other companies in this field?
You have to be better. Okay. Technologically. I mean, the lobby power of GE or Siemens or, it's not comparable to ours. We don't do lobby. In the US, I would know about it. So, and we don't. So it's you have to find a niche, and preclinical is one of the niches because for it is really R&D heavy, and I could see you a few applications. There are a lot more. So everybody is interested in their own molecule, their own research. So they want a company that has a lot of experience, but also can act quickly and understand their needs. So Merizo is ideal because it's large enough, we have about 350 people, something like that, globally, large enough to be robust and stable and profitable over a long period of time, while unlike GE Healthcare or Siemens, they have a really high overhead on R&D. Because of the structure of the company. Typically, large companies don't innovate; they buy the companies who innovate. So they do the technology transfer that way. So preclinical is quite a good niche for Merizo. Clinical is relationships, so that's important. So the sales process itself, because these equipment last for a decade and a half. So you have to build a good relationship, make people comfortable with you. Some countries they are more relationship based. Some countries are more meritocracy or technology based. And also we have a product nobody else has. It's a triple-headed SPECT CT. With PET we don't compete with the big guys because their IPs protect their technology. But we have a niche product on the SPECT CT market using the technology we developed for the preclinical and then scaled up to the clinical. So there's a translation in terms of technology, not just, as I mentioned in one of the slides, we started with a human camera and then went down to the preclinical field. And you remember 2,000 times larger volume. We also did the transfer the other way around. So we created something great preclinically and then scaled it up to humans. And large companies, because we don't have a preclinical, they didn't have that technology as fine-tuned. So that it's not easy. It's not easy, but it's feasible. And it's one of the messages I wanted to say is it is feasible to compete over a long period of time, to break into the market. When we broke into the preclinical market, Siemens and Philips were involved, and we still could make it as a success. And now we are market leaders in not all modalities, but front-end PET-CT in the US as well. Uphill battle, but it's possible.
So do you plan to get into the clinical business in the US? Do you have the long-term plans to do that?
Maybe with the researched triple-headed camera, because if we have a very similar offering to Siemens or GE, it's just fairly easy to push out the competition by lowering the prices till they just suffocate them basically, and then you raise the prices back to that's how it works. But if you find a niche where you can beat them, yes, so it's there's a plan. We have some of these products are FDA approved, and we have clinical installations in the US as well. Not the large triple-headed SPECT-CT is not yet FDA approved.
There's another remote question. Is that an employee perk at the Mediso HQ in Budapest for colleagues to get scans and check if they have any dysfunction?
Oh, fantastic question. Actually, I tell you this story. It happened with a person called Zsuzsa. Zsuzsa was, without obviously mentioning the surname, was gaining weight quite a bit, and like very rapidly. Didn't change, eating habits or whatnot. So the doctor sent her to an MRI to check for a brain tumor. And every MRI, after every MRI, you get the CD or DVD, and then the doctor said, "Oh, it's a negative, so there's no brain tumor." So they were developing this piece of software I showed you, Interview Fusion, and somebody came to the office asking for a MRI, because back then we didn't have MRI. And she was like, a couple of weeks ago, I had a brain MRI. So brought in the CD, tested the software, brought up the brain of hers, and found the brain tumor actually, because the doctor who did the diagnosis didn't check every part of the brain, so the brain tumor was not exactly right there. That was responsible for the weight gain, but a little bit off, still pushing the same region. So had to do surgery. She's still around, working with us after like maybe 10 years ago or so. So there are some perks. And within the Mediso group of companies, there's also several clinical centers now in Hungary. So I hope nobody will ever need that cancer diagnosis among my colleagues.
I just one more question. So what are the latest technological advances in this field? What your company is focusing on these days?
So one of them in the PET clinical field, it's a great question, in terms of technology, is scaling up these cameras. So typically a PET scan covers historically about 15 or 20 centimeters, these days about 30 centimeters. But to have a 2-meter PET that covers the whole body in one go, so you don't need to move the patient in and out over time. So usually how it works is you have a section and the patient moves, another section scan, the patient moves. But you can't see the whole body simultaneously. So this is why this video was pretty good, showing that you see the whole body in one go. So it is very expensive still, so it's about $12 million or something like that. But it's feasible because the scintillator technology has not changed too much, but how we detect the light coming from the gamma rays, we have a wafer technology since maybe 2008 or 9 when it started doing it. It's much cheaper than a photomultiplier tube, because a photomultiplier tube is literally like an old TV, just smaller. It's a vacuum tube. It is very sensitive. It it's a piece of glass essentially. But with the wafer technology, you can still go down and build some of those big ones. So it is funded by a Chinese company, United Imaging, and worked with UC Davis. So that's one on the instrumentation side. In terms of the spec side, it is how to increase the number of heads. So this is why Medizo has 3 heads, for instance. In terms of CT, it's like more dual energy and all sorts of other things. MRI, it's higher field, new tests. So it is still a dynamic field because people don't want to suffer, people don't want to die. So healthcare is still very lively, and imaging is an unprecedented tool for diagnosis. So it's there is innovation a lot. From the main, like the basic component to software, and AI is one of them. I'm sure everybody's by now a bit bored of AI. So, for instance, a couple of years ago, NVIDIA was funding projects. So it's very easy to create these images, but it takes time to read them for a doctor. So usually it's on a first come first served basis. So doctors read the images. Mammography, for instance, it's very easy to do and very quick to do. It's a routine, but the bottleneck in the process is the doctor's time. So one idea is to have the AI going through all of these images and flag the ones up that might show some abnormality, some disease, some it's a nice word, but I forgot. So one thing to, instead of if your exam was taken 3 months ago and now it's that your time for the doctor to look at the image, to have a priority list, and the AI suggests the doctor to read that image first because it might be more interesting.
So it won't be too long before the AI will make a diagnostic decision.
Yeah, we are going that way. And also we use AI for MRI noise, because you don't want too much noise in the image, because small details might just get buried in the noise. It's like speaking very softly, you might not hear everything. It's similar in imaging. So to improve the signal to noise with AI and some other algorithms, but the first one is easier to understand and can be used in clinical practice.
How do you see cost, especially coming down in order for early screening or early detection? I think cost is typically prohibitive for those sorts of applications.
So great question. In terms of the nuclear techniques, typically nuclear techniques are not the first one people use. So you have to have some sort of a symptom, or at least some suspicion that something is off, because they are, a bit cumbersome. Still very powerful, but you have to inject the patient, certain procedures, and you don't want to give patients radioactivity just for the sake of it. I work for other companies, and pretty much in every country it is available, from India to Pakistan. I work for an Israeli company, spec cardiac product, and maybe I shouldn't say because there's an embargo, but anyway, so they got at the end what they wanted. So it is fairly available, not for everybody, and not all of these modalities. So PET is a rich person's modality because it's very expensive to create those radioactive molecules. SPECT is much simpler. So it's quite widely available. I think we have Zambia as well, Zimbabwe, many of the African countries. They are not tomographic, so you can't, they're just one slice. But for instance, a thyroid disease or a kidney disease, you don't need to have a tomographic way. It's better, but you don't need. With the funkier stuff, with Alzheimer's and Parkinson's. I worked with the South African government at some point, and the people in South Africa are envious of their fellows who have cancer, because cancer is the rich man's disease. Because if you are not rich, you died from malaria or AIDS or one of those already. So it's a very interesting thing. We are not to do very well TB, tuberculosis anymore, but in some countries it's still a huge issue. So if you think globally, there's still a lot to do. Obviously, if you live in a Western country, the US, you have access to the newest technology always. It's not true in every country.
And what about side effects for screening and early detection?
Negligible at that point. So you have, that's another thing I could have touched upon today, of how much dose you give and what sort of dose is equivalent to riding a motorcycle, or like crossing the road without turning your head left and right and checking whether a car is coming or not. Typically not much. Like if it's ischemia or myocardial infarction, you don't really worry too much about the dose, because that's the lesser of evil. But some cases for kids, for instance, low dose is a biggie. For a sick kid, for instance, leukemia, or somebody who is already quite vulnerable, and CT is a bigger issue.
CT is a big one, yeah.
Like much more than PET or SPECT.
There was another remote question. Whether can you take standard MRI data and read it with your software?
Absolutely.
Is there much use of VR headsets in the interpretation of the imagery?
I can tell you from experience that doctors love their own display. So sometimes they want to keep them for even decades. Because, it's like Photoshop, seriously, it wasn't a joke. It depends on how you set the window of the visualization. You can see very different things. It's just like when you take your camera out and take a photo of somebody or something, and add a few filters, they look very different, 20 years younger or older, depending on what you want to see. Doctors love their own displays and their own color scales, because they train their minds over a long period of time to judge whether somebody is ill or not based on the images, based on a certain color scale, based on a display. It's a lot of responsibility. So maybe the future doctors who use VR or any of those technology visualization, advanced visualization, but it's critical. Some people only do grayscale, because when they were young, they started using grayscale monitors. I had one of those customers, Royal Brompton Hospital in London. And he was one of the best nuclear cardiologists ever. His brain was trained to read MIBI, or cardiac images, so well. So sometimes innovation is great. At the end, the doctor makes the decision.
Thank you very much. Thanks everyone for coming, and please remember to check out our website at emsv.com, and please join us for some